Okay. Hello. So today I'm going to discuss about some question on the cell basic unit of life. So okay, let's begin with so s- so let's begin with the first question. So the question is from previous year questions and last 38 years, and the chapter is 8th chapter, and the cell the cell and the basic unit of life. Let's move to the okay, first question: which of the following is the smallest cell? So 4 options are given. You have to choose the correct answer from this question. So first option A, virus; option B, acetabularia; and PPLO, pneumonia like organism; and chlamydomonas. So the PPLO, pneumonia like organism, now known as mycoplasma, are the smallest known cells capable of replications. They lack a cell wall and can be as small as 0.1 to 0.3 micrometers. Viruses are acellular and not considered true cells, while acetabularia and chlamydomonas are larger eukaryotic cells. So these are the explanation for this question. So let's move to the next question. So next question is the pneumonia like organism were discovered by. So four options are given. You have to choose the correct answer. Who are watching this video, you just try to answer on the comment section. I'll just 30 seconds question. So this is the question: the pneumonia like organism were discovered by knockout and rocks. So the PPLO, pneumonia like organism, now classified under mycoplasma, were first discovered by knockout and rocks in 1898, while studying bovine pneumonia. Their work laid the foundation for understanding this unique bacteria. So let's move to the next question. So next question is the term was introduced by. And so 4 options are given. So option A is Robert Hooke. Option B, Darwin. Option C, Robert Brown. And option D, Alfred Wallace. So the correct answer will be the Robert Hooke. The Robert Hooke introduced the term. The first cell was termed Robert Hooke. The Robert Hooke coined the term cell in 1665 when he observed the honeycomb-like structure of corpse under a microscope and published his finding in Micrographia. So he published his work in a journal which is called as a landmark work in the development of cell theory. So this is a very good question, and let's move to the next question. So question number 4: figure of cork cell observed by Robert Hooke were published in. So I just a little earlier I have told you that when he found or discovered the cells, he published his work in Micrographia, which is a journal. So Robert Brown Robert Brown's detailed structure, observation and illustration of cork cells were published in the book Micrographia in 1665. This work was instrumental in introducing the concept of cell to the scientific community. So the correct answer is the option B. Let's move to the next question. The cell theory was given by. So cell theory was given by Sladen and so on. So the cell theory states that the all living organisms are composed of cell and that the cell is the basic unit of life was formulated by Matthias Sladen and Theodore Swan in 1939. So the theory was later expanded by Rudolf Virchow, who introduced the idea that the cell arises from pre-existing cells. So let's move to this is the explanation for this question. If you like the question, so if you like the question and answering sessions, though you can like the video and share with your friends. Let's move to the next question. The major breakthrough in the studies of cell came with the development of electron microscope. This is because so 4 options are given. The electron microscope is more powerful than the light microscope as it uses a beam of electrons which has wavelength much longer than the photons. And option B, resolving power of the electron microscope is much higher than that of light microscope, and the resolving power of electron beam is 200 to 350 nanometer as compared to 0.1 to 0.2 nanometer for the light microscopes, and can pass through thick material where light microscope require thin sections. So the correct answer will be the option B. The resolving power of electron microscope is much higher than that light microscope. So let's move to the next question. The student wishes to study the cell structure under a light microscope having 10x AP eyepiece and 14 and 45 the object by which one of the following colors of light so as to get the best possible resolution. So the correct answer will be the blue. So let's move to the next question. The cell theory stated that 4 options are given. All cells are living, all cells possess nuclei, all cells reproduce by mitosis and meiosis, and cells are fundamental structural units. The correct answer will be the cells are fundamental structural units of plants and animals. So the correct answer is option D. So explanation are given. I'm not going to discuss about the explanation in details. If you want PDFs, contact me in the comment section the PDF for this question. And if you like my question answering video, then you can offer me a cup of tea. So let's move to the next question. Omnis cellula e cellula. All cells comes from cells. So the above statement was given by. So 4 options are given. So the correct answer will be the Rudolf Virchow. So Rudolf Virchow proposed that the principle of omnis cellula e cellula in 1855, meaning all cells arise from pre-existing cells, completing the cell theory. The Robert Hooke, so other option is Robert Hooke is known for discovery of nucleus, and Sladen is contribute the cell theory by stating that the plant cells are made up of cells, but he did not propose omnis cellula e cellula. And Theodore Swan extended the theory to the animal, but the specific statement is attributed to the Virchow. So the correct answer for this question is here the Rudolf Virchow, who stated that the omnis cellula e celluli, the above, so pre-existing the cell, all cell arise from pre-existing cells. So this is the third statement of the cell theory. Let's move to the next question. The cell theory is not applicable to. So 4 options given: algae, viruses, fungi, and lichens. So So the correct answer will be the cell theory is not applicable to the viruses, because cell theory applies to the only living organism made up of cells, but viruses are acellular. They are not cells and considered as a non-living cell outside the host. The reason the cell theory is not applicable to the virus, but the others like algae, fungi, lichens are cellular organisms. That's why the cell theory applied to the algae, fungi, lichens, but not to the viruses. So let's move to the next question. The main differences between animal and plant cell is that. So main differences between the plant cells and animal cell. So option A, plant cells lack a rigid cell wall. Animal cell lack rigid cell wall. Option C, plant cell possess small vacuoles, and option possess large vacuoles. So the correct answer will be the option B, animal cells lack rigid cell wall. So the explanation is given here. So if you want to PDF for this session, so let's move to the next question. Which of the following statement is not true? So for option are given animal cells. Option A, animal cells never contain chloroplast unlike many plant cells. Option B, plant cells have cellulosic cell wall while animal cells do not. Plant cells have central vacuoles while animal cells do not. And option D, animal cells contain microscopic chloroplast. So the correct answer is option D. The animal cells don't have any kind of chloroplast bulb. So that is the main statement which is false. Is not true about the animal cells. So option D here will be the correct answer for this question. Let's move to the next question. So here I hope the statement is false, because animal cells don't have any kind of chloroplast. So animal cells don't have any chloroplast, while the plant cells contain chloroplast, which is microscopic for photosynthesis. And ultrascopic and microscopic are non-standard in the context and seem to be misinterpretations. Let's move to the next question. The name of Sladian and so on are associated with. So protoplasm as a physical basis of life. Option B, cell theory, theory of lineage, and nucleus functions control center of cell. So the correct answer will be definitely the cell theory. So Sladian and Matthias Sladian and Theodore Swan formulated the cell theory in 1839, proposing that all plants and animals are made up of cells, establishing the cell as the fundamental unit of life. So, but other option like protoplasm as a physical basis of life. Who gave these statements? Who proposed this statement? Parkinson. This statement is associated with Parkinson and Huxley, not Sladen and so on. And theory of cell lineage, Rudolf Virchow. In the last third statement of the cell theory, Virchow added that all cells arise from pre-existing cells. So, and the nucleus function as a central control center of cell. So nucleus function as a central control center of the cells related to the Robert Brown's discovery of the nucleus and later research. So this is the main explanation for this question. Let's move to the next question. Magnification of compound microscope is not connected with numerical aperture, focal length of objective, local length of eyepiece, and tube length. So the correct answer will be the numerical aperture. Let's move to the next question. The angstrom, 1 angstrom is equal to 0.001 millimeter. So we know that the 1 angstrom is equal to the 10 to the power minus 10 meters, and if you convert it to the millimeter, will be 10 to the power minus 3. 1 millimeter equal to 10 to the power minus 3. So 1 angstrom equal to 10 to the power 10 millimeter equal to 0.0001 millimeter. So the correct option is option C, 0.001 millimeter. So let's move to the next question. Organelles can be separated from cell homogenate through. So the correct answer will be here for this question is chromatophore, option B X-rays diffraction, differential centrifugation, and autoradiography. So the correct answer will be the organelles are separated by differential centrifugation, because so differential technique is used to separate cell organelles in cells, and chromatography is used to separate different type of proteins, and X-ray diffraction is used to study the structure of molecules, and autoradiography also used to detect the substance in a sample. So this is the explanation for this question. I hope it will help you. Let's move to the next question. Which of the following statement about inclusion body is incorrect? So inclusion body, 4 options are given. You have to choose the correct answer from this question. They are, option A, they are not bound by any membrane. These are involved in ingestion of food particles. They lie free in cytoplasm, and these present reserve material in cytoplasm. So the correct answer will be that they are involved in ingestion of food particles. So here, inclusion bodies are non-living structures found in the cytoplasm of cells, typically consisting of materials such as glycogen, lipids, protein crystals, and they are not bound by membranes and lie free in the cytoplasm and serve as a storage for reserve materials. So this is the, so for this question, that here the question asks you to find out which one is the incorrect about the incorrect statement about the inclusion body of. So inclusion body is they are not involved in ingestion of the food particles. So option B is the correct answer for this question. Let's move to the next question. Select the wrong statement. So 4 options are given. So you have to find out the wrong statement. It has come to the NEET exam 2016 of peptidoglycan. Option B, pili and fimray are mainly involved in motility of bacterial cells. Cyanobacteria lack flagellated cells, and mycoplasma is a wall-less microorganism. So the correct answer for this question is pili and fimray are mainly involved in motility of bacterial cells. No, pili and fimray are structure on the bacterial cells that primarily function in attachment and adhesion, not motility. So motility in the bacteria is mainly achieved through the flagella. Thus the statement is incorrect. Why? The bacterial cell wall is made up of peptidoglycan. It is a correct statement. And cyanobacteria lack flagellated cells. It is also a correct statement. And mycoplasma don't have any kind of cell wall. So it is also a correct option. So let's move to the next question. Which one structure is not found in prokaryotic cells? So 4 options are given. You have to choose the correct. So option A, mesosome. Option B, plasma membrane. Option C, nuclear envelope of ribosome. So the correct answer for this question is option Nuclear envelope is not found in prokaryotes because the prokaryotic cells lack nuclear envelope, which is characteristic feature of eukaryotic cells that encloses the nucleus. So prokaryotics have a nucleoid region instead of the nucleus. So the mesosome, which is found in the prokaryotics, it is involved in cell wall formation and respiration. And plasma membrane present in all cells, including prokaryotes, and ribosome present in prokaryotic as well as eukaryotes. So next question, let's move to the next question. So the structure that helps some bacteria to attach to rocks or host tissue are. So four options are mesosomes, holdfast, rhizoids, and the femri. So the correct answer is definitely will be the option D, femri. So femri is a femries are hair-like appendages on bacterial surfaces that help in attachment to the surface, such as rocks or host tissues. And other option like mesosome involved in respiration and cell wall formation and not attachment. And holdfast, a structure in algae, not bacteria, for attachment. Holdfast is present in certain kind of algae, and it is used for attachment. So it is not present in bacteria. As well as option C, rhizoid, which is found only in fungi and plants, not in bacteria. So the correct answer for this question is femri. Femri is the appendages, hair-like appendages on the surface of bacteria that help in attachment to the surface such as rocks or host tissue. So let's move to the next question. Which one of the following is not an inclusion body found in prokaryotes? So which one of the following is not an inclusion body found in prokaryotes? Option A, glycogen granules. Option B, polysome. Option C, phosphate granules. And option D, cyanophycean granules. So the correct answer for this question will be definitely the polysome. So polysome is not an inclusion body found in prokaryotes. The polysome is a complex of multiple ribosomes translating mRNA, not storage. It is not involved in storage any kind of food materials. So inclusion body in prokaryotes include glycogen, phosphate, and cyanophycean granules, which store reserve material. So other options. So the correct answer here option B. So let's move to the next question. Which structure perform the function of mitochondria in bacteria? So very easy question. You can easily question. The mesosome will be the correct answer for this question. So mesosome are involved infoldings of bacterial plasma membrane which perform function similar to the mitochondria. It is involved in respiration and ATP production. As well as it is also involved in cell wall formation. So it is not a true mitochondria. It plays the role like mitochondria in bacteria. So mitochondria, prokaryotes don't have any mitochondria, but they have plasma membrane infoldings which are involved in respiration. So the nucleids. So the correct answer for this question is the option D. But other option, nucleids contains bacterial DNA, not involved in energy production. And ribosomes involved in protein synthesis, and cell wall provide structural support, not involved in any kind of metabolic function. So for this question, the correct answer will be the mesosome. So let's move to the next one. The motile bacteria are able to move by. Very easy question. I have already discussed similar type of questions in the previous questions. So motile bacteria are able to move by. So option A, fimri. Option B, flagella. Option C, cilia. Option D, pili. So the correct answer for this question is definitely will be the flagella. So flagella are whip-like structure that enable motile bacteria to move from one place to another by rotating and provide and it's help in the locomotion. The fimri are used for attachment. Cilia is found in eukaryotic, mainly protozoa. Cilia like paramecium. Cilia is not found in bacteria, and pili involved in attachment and DNA transfer, not motility. So the correct answer for this question is here, the flagella. Let's move to the next question. Why is a capsule advantageous to a bacterium? It protects the bacteria from a desiccation. It provides means of locomotion. It allows bacterium to hide from host immune system, and it allows the bacteria to attach the surface. So the correct answer for this question will be it allows the bacteria to hide from the host immune system. So the capsule is a polysaccharide layer that surrounds some bacteria, helping them evade the host immune system by masking antigens, thus aiding the survival. So here the correct answer for this question is option C. Let's move to the next question for this. So the next question is here, the term glycocalyx is used for. So glycocalyx is used for. So option A layer present between cell wall and membrane of bacteria. Option B, cell wall of bacteria. Option C, bacterial cell wall, glyco-engineered to possess N-glycosylated protein. And option D, a layer surrounding the cell wall of bacteria. So the correct answer for this question will be the option D, a layer surrounding the cell wall of bacteria. So glycocalyx is a polysaccharide layer cell wall of bacteria which provide protection and aiding in or help in attachment. So it can be a loose sheath or well organized capsule. So let's move to the next question. So the chromosomes in a bacteria cell can be 1 to 3 in number and are always circular, are always linear, can be either circular or linear but never both within the same cell. And option D, can be circular as well as linear within the same cell. So the correct answer for this question is are always circular. So in bacterial cells the chromosomes are always circular. So the bacterial chromosomes are typically single circular DNA molecules, though the number can ranges from 1 to 3 in some species. So linear chromosome are rare and not in normal in bacteria. So the correct answer for this question is here option is bacterial DNA are always circular. So remember this. Let's move to the next question. Photosynthetic bacteria have pigments in option A, chromatophores, chromatoplast, chromatophores, option C, leucoplast, and chloroplast. So you know that bacteria, photosynthetic bacteria have a special structure which is called chromatophores that help in photosynthesis. The chromatophore helps in photosynthesis in bacterial cells. So bacteria don't have any kind of plastids or chloroplast or other type of plast, but they have a pigment type which is called chromatophores, which is a specialized membrane bound structure that house pigments like bacteriochlorophyll. So bacteriochlorophyll is the pigments that help in photosynthesis or to make the food. So let's move to the next question. The site of respiration is in bacteria is. So site of respiration in bacteria is. So mesosome will be the correct answer. I have already discussed. The mesosomes are enfoldings of plasma membrane in bacteria that serve as a site of respiration, performing function of similar to the bacteria in the eukaryotic cells. So ribosomes not involved in the respiration, it is involved in the photosynthesis. Microsomes, an artificial structure from fragmented endoplasmic reticulum, not site of a respiration. And the plasma membrane genetic elements, episomes is a plasmid which is present in bacteria, and it is not involved in respirations, but it is involved in genetic of bacteria. So let's move to the eukaryotic cells. So I'll discuss some question from eukaryotic cells, but I just want to tell you that if you like my question and answering, so you can like my video. And so in the next video I will discuss about the question, so which is very so next question I will discuss about the eukaryotic cells. Question about the eukaryotic cells. So let's end here today's